Hi, I'm Gretchen, a 50-year-old living with familiar partial lipodystrophy. I do not have an exact type. My gene mutation is still unknown, but I do have all the usual perks that comes with lipodystrophy, like diabetes, insulin-resistant, fatty liver, chronic pancreatitis. I am also fortunate enough to have some autoimmune disorders, including Crohn's disease. After a bowel resection that removed 1.5 feet of my intestines, I've been in remission by using Humira. Humira is a biologic med used to treat several autoimmune conditions by reducing inflammation. My journey through life has a few major turning points that I would like to share today. I can remember being at work on New Year's Day 2015 and complaining that I wasn't feeling well. After my shift, I drove to the local hospital to find out if I was having some kind of Crohn's disease flare. I remember my husband bringing me my charger, and that's when we found out I had pancreatitis. I was admitted on a medical floor, but things escalated so quickly. I do not recall much. I remember laying in the hospital bed screaming that my chest felt like someone stabbed me in the heart. There were nurses and respiratory staff hovering over my bed, trying to reassure me that I was okay. I went from that room to the ICU, where they were able to give me more pain medication since I was monitored at all times. When I woke up again, I was now facing kidney and respiratory failure. My nurse told me they could not perform plasmapheresis that I desperately needed. I was transferred to a trauma unit in Pittsburgh and received plasmapheresis later on that night. My triglycerides were 8,873. The first few days in the ICU are a blur. I remember on the first night I was telling my nurse she was being dramatic when she said I was very sick. She looked at me and said, "You are not out of the woods yet." And I can still hear her saying it. That was the very first time I felt worried for myself. My body was on fire. Head to toe felt like I was dangling over a volcano. My mom placed washcloths that sat in ice all over my body for hours, switching the lukewarm cloths for the cold ones. I even pleaded to God if he saved me, I would get the kids baptized, which I did. I was so sick. I spent 10 more days in the hospital due to being swollen, low blood count, and of course, I had to be able to eat without getting sick. That hospitalization didn't just affect my body, it affected my mind too. My therapist believes this was the first of many medical trauma and PTSD. For the next 5 years, I found myself back in the hospital with pancreatitis. It became a pattern I dreaded. The familiar pain, the test, the quiet worry about how much damage is being done to my pancreas, and would I ever end up on a transplant list. The second year I ended up in the hospital was supposed to be my granddaughter's first birthday party. My daughter canceled it, and I still feel guilty 6 years later. My heart broke for us as it did when my body let me down. My last hospitalization broke the dreaded pattern when a medical student suggested I go to the National Institute of Health. At NIH, Dr. Brown and her team looked at the whole picture. For the first time, everything connected. That was where I finally received my diagnosis of lipodystrophy. After my diagnosis, I wanted to do more than just survive. I wanted to be a part of something that might help others living with lipodystrophy too. I remember Dr. Rao calling me at 8:30 at night to discuss a medication trial. I decided to do the trial to actively participate in something that might change the way lipodystrophy was treated. After a few months of weekly injections, I could walk more than 3 steps without feeling like I was carrying 700 pounds. I wasn't gasping for breath the way I used to. My guess is that I had the medication and I was getting better. The trial medication was supposed to be available for participants to continue treatment after, but the drug company pulled that opportunity for me. I never heard from the drug company to explain to me why I was not able to continue using the drug. Before I knew it, almost 2 years had passed without any real treatment. It was frustrating. I tried to stay hopeful, but there were days when I felt like I was slipping backwards. I didn't realize at the time, but the disappointment from the drug company played a much bigger role in my mental spiral downwards. To go from hope to nothing. That kind of emotional whiplash doesn't just fade, it burrows in. It changes how you see yourself, your future, your worth. But this journey has not always been just about physical. There's been moments that have broke me mentally and emotionally. Turns out that just having the diagnosis really meant nothing here in PA. No one could help locally. In 3 months, I was diagnosed with flu, COVID, and then pneumonia. Much like every time I explain my medical history and why things are the way they are for me, I get blank stares, and one time I even got a loud sigh. I never know how much to say before they think I'm insane or that I'm there seeking drugs. At my lowest point, I cried every day for 6 months. Every health appointment I went to ended with a new diagnosis and medication. During a simple annual diabetic eye exam, I learned that I have macular degeneration. I reached a place of pain that scared me. I actually began having thoughts to stop fighting the diseases and just simply drift away. It didn't matter when or how. My plan was simply just going to stop taking medications. That's when I started therapy. I learned some things that changed my life. She taught me how to grieve the woman I was and to embrace the process of becoming who I wanted to be. Together we healed the little girl inside of me. I learned that I couldn't control what was happening to my body, but I could learn how to guide my thoughts. I learned compassion for myself. I learned how to breathe through fear. And I remembered who I was before all of this. I'm too tough to let lipodystrophy ruin me. I've stopped being the victim and became tougher than the disease. I also learned to stop hating myself for not being able to be the mom and Gigi I felt I should be. I found that I can clear my head every day simply by taking a walk at a nearby park. That summer, I came across a post on Lipodystrophy United that stopped me in my tracks. It was from a girl who was feeling down about herself, and the sadness in her words made my heart sink. I could feel the weight she was carrying, the kind of weight I knew all too well. She was beautiful, but she couldn't see it, and something about that hit me deeply. Maybe it's because I'd been there. Maybe it's because I knew all too well how hard it is to live in a body that doesn't follow the rules. I couldn't just scroll past. I felt this pull to reach out, to let her know she wasn't alone. I commented on her post, telling her all about myself, about being older, having fat pads all over my body, and all the things that lipodystrophy has shaped in me. And then I told her something simple but true, that she was beautiful, and that our bodies are not who we are. I told her I was 48 years old wearing a bikini for the first time ever. I was no longer hiding body parts that others might laugh at. It was about showing her that our bodies, no matter how different or complicated, still deserve sunlight, still deserve joy, still deserve to exist without apology. I received a message about attending the support group, Tea with Lipodystrophy United, and started to join weekly. I knew immediately I was with my people. There is something almost magical about meeting people who understand your world without needing a single explanation. It's like recognizing pieces of yourself in someone else. The struggles, the strength, the humor you use to get through the days. Those connections felt natural from the very beginning, as if we've known each other far longer than we actually had. I've learned so much from them, and not just about the condition, but about living in a way that still leaves room for joy. Tea with Lou became one of those small but powerful rituals that gave me purpose again. Sitting together, sharing stories, laughing about things only we would understand, it reminded me that community isn't just about support. It's about being seen. It's about belonging, and it's about finding comfort in the simple acts of being understood. Those friendships helped me rebuild parts of myself I didn't even realize were fading. They reminded me that in the middle of a rare and complicated condition, there is connection, there is warmth, there is a place for me. And that sense of purpose, the feeling of being part of something bigger than my own struggles, became one of the most healing parts of my journey. In a world where this disease is so rare, that kind of connection is everything. Today, my fight is not fueled by anger. It is fueled by love. Love for the community. Love for the people who came before me, like my father and my brother, who passed away without ever knowing they had lipodystrophy. And love for the future. I participated in clinical trials not because it's easy, but because we need answers. Advocacy doesn't always look loud. Sometimes it looks like showing up. Sometimes it looks like sharing your story. Sometimes it looks like simply refusing to give up.
Wow, that was emotional.
Thank you. And thank you guys, truly, for every week listening to my nonsense. It means the world to me.
Thanks for coming each week. I think we're going to skip on our mental health resource because of time. So Andrew, if you want to start your talk and introduce what you do.
Yeah, it's going to be a little hard to follow that, but I will try. So I just requested to share my screen.
You should be able to.
Okay. So I think I'm going to have to do this a little odd because I have a really big screen so that I can do a lot of data analysis. So it gets really weird if I share my whole screen. So I'll just have to do sort of this mode. Let me see if I can. Yeah. So, hi everyone. I am Andrew Lutkewitte. I'm very honored to be here and to talk about the research that I do in my lab at the University of Kansas Medical Center. We really are interested in metabolic health and how your adipose tissue regulates that. And we focus mostly on the adipose liver communication or crosstalk. So I have a very fancy title, and this would be a title that I would use when applying for a grant or giving a research talk, and it is Identifying Mechanisms by which Adipose Tissue Deficiency Leads to Hepatic Metabolic Reprogramming. And I know that's a mouthful, but basically, we want to know, why does your liver make fat when your fat can't? Okay. And so I have a couple of disclosures. I'm not a clinician. I'm a basic scientist. So I apologize for if it's a lot sciencey. And I also have no financial disclosures, but I would love some. So if you know any pharma company that wants to help me do the research that I love, send them my way. It's relatively early in my career. So hopefully I get the opportunity to keep going and get paid to do that. So this is what my lab studies. These are the actual members of my lab. So this is Pradeep, who I think is on the call today. He is my graduate student. He's very talented. He's really interested in using um, omic data or big data sets and computer analysis to kind of identify new pathways in lipodystrophy and obesity. This is Sharisha. She's my lab manager, and she's just probably the best person in the world. And then right now, it's just the three of us, but Senjin was a rotation student. And so I always try to take either undergrads or even high school students because I think everyone has to start somewhere. And if they can start in my lab, that's even better. Okay. So that's who supports me at the lab. This is who supports me at home. So this is my wife, Kelly, and my beautiful daughter. She's 14 months, Lucy. So we're kind of working on hats and bows, but she's kind of a handful now. So as I said, I work at the University of Kansas Medical Center. That's located right here, kind of in the heart of America. I'm originally from St. Louis. My wife's from Cincinnati, so we're very Midwesterner. But I was recruited here to work with the Metabolism and Obesity Research Center. This is actually my lab right here. It's not the big one, it's the little one, but we have a really nice view. And I really need to kind of get them to change this from obesity research to maybe lipodystrophy and obesity research, right? And so I'm just going to jump real quick right in. And so this is adipose tissue. This is what it looks like on a microscope slide. This is actually from a mouse. We do mostly mouse models in the lab. We have some human studies, but these are actually different adipose depots. So this one is visceral or interabdominal, which would be here in your abdominal cavity. This one's subcutaneous. And I don't really want to spend too much time on this because many of you know that adipose does more than just store lipids, right? It also secretes various proteins like leptin, for example, that regulates your metabolism. And this is not my data, but I think it's really cool. So I always like to tell this story when I talk about adipose tissue. And so we often wonder how old is an adipocyte or a fat cell? How long is the lipid stored there? And so a very smart group of scientists learned that in the height of the Cold War, when all these nuclear bombs were going off, that was releasing radioactive carbon into the atmosphere. And this carbon gets incorporated into triglycerides, right? The fats that the mechanism for storing fat. Well, you can actually take people born before this and after this and figure out the difference in radioactivity. And so, if you're ever curious, the lipids in your fat cells last about 2 years. And in an individual, your fat or fat cell can be about 10 years old. And so I always think that's kind of interesting. Again, this isn't a lean or people with obesity. We don't know in lipodystrophy, right? We don't know how that changes. But I always think this is kind of an interesting thing. Many of you know that adipose stores lipids, right? It uses insulin secreted from your pancreas to convert glucose into fatty acids. And this is, like I said, under a normal condition. But actually, if we use a high-fat diet to induce obesity in mice, the adipose changes quite a bit, right? It becomes different sizes. All these little blue dots in there, those are actually immune cells. So when you hear inflammation, that's actually what we're seeing here. And they often call them crown-like structures. But lipodystrophy, so this is actually from my mouse model that I'm going to talk about here in a second, that lacks adipose tissue. It's almost worse, right? You have a lot more inflammation, you have a lot more fibrosis. And so, I know you guys probably get tired of being compared to, the obesity epidemic because it's a different disease. But I think that's why my lab is interested in it and how I got into it. Because lipodystrophy and obesity have the same sort of metabolic diseases, although lipodystrophy tends to be worse. And so it's really the quality of your fat tissue, not the quantity, right? And so that's kind of what we study. And we use mouse models to do that. We're really interested in understanding how your liver makes new lipids or how you get steatosis and fatty liver from different sources. And we use mouse models. So I actually spoke with Dr. Brown not too long ago, and this is from one of her papers. It's a little outdated, but these are the current therapies and treatments available, and I've kind of starred metronidazole. And there's a lot of them, but not everyone works for everyone, and not everything. It's not a cure, right? It's a treatment. And I added this slide, which some of you may recognize. The GLPs are working quite well in some patients with lipodystrophy. I actually am partial to the GLP therapy because I personally lost 50 pounds taking one. But it was not easy. It was not fun. I had a lot of side effects, and a lot of people can't take them forever. I could not. I basically had to stop taking it because it just made me feel awful. And so we are really looking for sort of new therapies or different ways to treat some of the conditions in lipodystrophy, right? Why does your liver make fat when your fat can't? And it does that through a process called de novo lipogenesis. That's a fancy term for saying glucose, which is a carbohydrate, gets converted through a big process in the liver to make fat. And fats are usually, palmitate is a name of a fat, okay? So in lipodystrophy, we know that your liver actually converts glucose into fats. This is data from a clinical trial or a clinical study showing that in lipodystrophy, at least 10% will say of your lipids in your liver come from glucose. And so, how can we inhibit that to maybe see if we can alleviate steatosis? We know in obesity this happens, and this has been known for a long time that in patients with high liver fat and obesity, the difference in that liver fat comes from new lipids. It doesn't come from your meals. It doesn't come from your adipose. You actually make more in the liver. And that's kind of how I got involved in this. So how did I get here? We at the time in my postdoc, I studied an enzyme called lipin-1, and that is involved in triglyceride synthesis, right? And triglycerides are how you store your fat in your adipose. So it's really important. And we were working in sort of the obesity field, and we were working with a group that was really interested in how some patients who suffer from obesity are otherwise metabolically healthy, so they don't have diabetes, they don't have any of those symptoms. But how is that so? So we actually looked in their fat tissue and showed that lipin-1 decreases with obesity and metabolic disease. Okay, but that's really important because in these same patients, they make more lipids in their liver inversely to lipin. And that kind of makes sense because if you can't make fat in your fat, your liver is compensating, right? So the more of this lipin you have in your fat, the less your liver is making on its own. And so, because I am a basic scientist, we made a mouse that can't express this protein in its fat. And I will say that lipin does have a human mutation. It does not result in lipodystrophy. It actually results in a condition called rhabdomyolysis, which is a fancy term for, I guess, muscle wasting. And that's because there's tons of different lipins in humans versus mice. But we just use it as a model, and it's a really good model. Our mice don't gain any weight on a high fat diet, so the wild types gain weight, the knockouts in red don't. And it's all different kinds of depot. So this is visceral or sort of belly fat, and this is hip fat. So they're really lean, and a lot of people say, oh, awesome, let's block lipid and fat, but you need fat, right? And so what ends up happening is they get really high insulin, and they get really glucose intolerant, right? So even though they're lean, they have glucose intolerance, they're insulin resistant. And what happens to their liver is it gets big, right? They have hepatomegaly. And I know that's a condition often seen in lipodystrophy. And so they get these really big lipid-filled livers, and these little white spaces are lipids in their liver, right? But I'm interested in how that happens. So we directly measured lipid synthesis using fancy techniques to show that these mice actually make more fat in their liver. And that's kind of was kind of a big deal because normally high fat diets suppress liver from making fat because if you're eating fat, you don't need to make it, but they maintain it, right? So it's this persistent, upregulated new lipids in the liver that I think is really driving a big portion of the disease. And so we published part of this project, but what we do is we take samples of their liver or tissue, and we use these fancy machines that is called mass spec, and we do proteomics. So we figure out every single little protein that's in the liver and how they interact with each other. And so we get these big computational models that sort of show these big mess of networks and stuff that doesn't, really make sense, probably to some of you, but it has given us actual targets to test to say, can we block some of these proteins and alleviate steatotic liver disease? And so, a lot of them actually exist, and you might recognize like metformin and rapamycin. Some are cancer drugs. We are really excited about some of these other inhibitors, and there's a lot of drugs in this space, and there's a lot of them that have failed for fatty liver disease. I think there's only one specific to fatty liver disease. And a lot of them that block lipid synthesis in the liver, they cause your plasma triglycerides to skyrocket, which is not good, right? So we are excited because we're going to try one that we think is promising. It's been shown in obese and in mice to work really well, but it's never been tried in lipodystrophy. So that's really what we were trying to do is discover new or novel targets. And
Sorry, Andrew, can I interrupt really fast
yeah,
and
yeah,
just see if anyone has any questions?
yeah, this
So
is actually a good stopping point. Sorry, that was kind of a lot.
it's a lot, but it's also really good information. But I want to make
Okay.
sure
I was
everyone.
hoping to kind of be able to explain it a little bit because I don't think people are going to like my next portion here, and I'll explain why.
Is
Okay.
anyone
Well,
confused or have any questions?
I can keep going a little bit, maybe.
Perfect.
Okay. Um, and so one of the things that I noticed in doing like literature reviews and doing all the patient treatments and reading all that, exercise shows up quite a bit, right? And exercise is very interesting to me because there's not a lot of clinical trials on exercise and lipodystrophy. There is on HIV-induced lipodystrophy, but not on any of the other conditions. And I know what you're saying. Ain't no way I'm going to exercise, or maybe I can't, and it's not necessarily a good treatment. And again, remember, I'm not a clinician, I'm a scientist. I'm really interested in this kind of aspect because in the exercise field, exercise is good for just about everything, from neurodegenerative diseases to cancer to diabetes. And if you notice, all of it has to do with adiposity and adipose tissue. It actually makes your adipose tissue healthier. And the way it does that is your muscle, when you increase your activity, causes the liver and adipose tissue to release fuel so that you can exercise. So in obesity, exercise will pull lipids out of the liver so that you burn them in skeletal muscle. Um, and I'll get to that in a second. So we can exercise mice. It doesn't look like this, actually. It tends to more look like this. And so this is the center that I went to. They have some of the best mouse metabolic equipment in the country. We actually have little disc wheels. So you can put a mouse on here and they will run and run and run, and we can count how long they run. We have treadmills. So each one of these is a lane. You can put a mouse in there. You can do inclines and all this stuff. These are super fancy. These are treadmills that, if you've ever heard of like athletes having VO2 max or like maximal expression, we can actually measure that in a mouse. And so the thinking is, or sorry, so mice are incredible. They run 7 to 20 kilometers a day, which is just insane to me. This should actually say at night because they're nocturnal. They typically run at night. So they will run. But the question is, can someone, or can a mouse that doesn't have adipose, doesn't have fuel, can that run? Is this even a viable option, right? Is this silly? Should I just be slapped, right? Especially when you don't have adipose to support your muscle, you don't have these fats in at least in this model. And it turns out we were able to run these mice. So the lipodystrophic mice run just as much as the wild type. Female mice tend to run more. And so this is all pilot data, just to see if it's feasible, because I'm currently writing a grant for the American Diabetes Association on this. Um, they tend to eat more when they run to fuel that. And we use surrogate markers of liver health in these models. So Splenomegalia is often an indicator of portal hypertension. So in the sedentary mice, they have big spleens. That's always true in lipodystrophy. When they run, it kind of rescues that, right? And these are just inflammatory markers that seem to go up with lipodystrophy. Exercise restores it. So I'm not saying that this is the treatment because I put this data together this week, so this was very last minute. So one of the things that really shocked me and some of the other people I work with who are exercise physiologists is running or exercise actually increased their plasma triglycerides. And it actually increased their liver triglycerides, at least in the female mice. So maybe it's not something to do, right? Maybe there's more to it than just exercise is good, right? Because, if we go back to like the theme is why does your liver make fat when your fat can't? Maybe this isn't a good thing, right? And so this is kind of what basic scientists have to deal with on a daily basis, right? You have this thing in mind, and then it tends to be the opposite. In fact, if we look at all the different proteins or enzymes involved in lipid synthesis, they actually are all upregulated with lipodystrophy and exercise. And so maybe you were right. Maybe exercise isn't the answer, but there is something that is kind of interesting that's called, I don't know if you've ever heard of this. It's called the athlete's paradox. So professional athletes actually get more lipids in their muscle tissue and some of their other organs, but they're more efficient at burning it. And so their machinery is better. So maybe that's what's going on here. But the question was really, in the context of not having adipose to support that, maybe it's a way to lower blood glucose, but maybe it's a way to just improve health overall. But if you can't do it, right, it doesn't matter. And how am I for time? I think I got 10 minutes. Okay. So I threw this in last minute because I do a lot of different things in the lab. I have another mouse model that is something that I am kind of fascinated by. This mouse is called, you might have heard of it before, it's called a fat-free mouse or a fatless mouse. So we can actually um, have this, we can inject a toxin into this mouse and it will kill their fat cells. And then we can actually monitor their health going forward. So it's a way to acutely sort of injure or affect adipose or create adipose dysfunction, and then we can monitor their metabolism. In a very short period of time, they lose, this is it's gut fat and this is subcutaneous, so they lose quite a bit of fat in that short period of time. But they completely lose their adipose function. So if you remember leptin, that's secreted from adipose. They pretty much can't secrete that. They don't have adiponectin at all. And so what ends up happening is they get super high blood sugar and they get super high insulin. So we're kind of using this mouse as a model to test various drugs and various things on how the liver responds to not having adipose tissue. Because just like in the other model, they have hepatomegalia, so they get a big liver, and then they get lipids in their liver. And so I have a lot of projects going on this. And I know that was kind of overwhelming, and I hope that you learned a little bit of something, and I can just um, maybe show that there are, maybe if the current therapies aren't working, give it time. We're still kind of working. And again, my theme is really how does your liver make fat when your fat can't? We think we've identified some targets, and we can hopefully apply them to both lipodystrophy and obesity. I didn't have time today to talk about. We have a very large catalog of human samples. I'm starting my own adipose biobank, and so we're learning a lot in that too, through a lot of different techniques. So, with that, I guess I will. I do have acknowledgments. The people that I work with at Kansas University Medical Center. It's a really great place. I like it. It's a very small but up and growing institute, and I'm trying to spread the word of lipodystrophy. And I noticed you guys have swag, so I am totally buying some shirts later after this to wear as being a lipodystrophy warrior. So with that, I will take questions.
Great, thank you so much. Do any of you have any questions for Andrew?
Can I quickly ask a few
Yes.
questions? Just,
Yeah.
thank you for the interesting presentation. So, do you was running studies, the running studies that you've done, were they voluntary running or
Yes. So I didn't, yes, they are. It's this little disc. We put it in their home cage and it's all voluntary. So they just run whenever and how often they want. So the follow-up study, and you will kind of understand this, is we're going to put them in indirect calorimetry systems with wheels, but I need money to do that. But it was very interesting that they did run, and I've only ever found one other study in the CPIN mutation that showed that they also run, but it was very limited. And
we
so,
recently published our laminate
yeah,
data
yeah,
in JCI, and that I mean the part of this study that hasn't been published yet on multiple knock-in variants.
hmm
Um, th- um, we have like 7 different knock-in variants in
hmm
mice, and we seem to find, at least in the mice that harbor the laminae variants, that voluntary running is reduced. But
hmm
if you actually put them on a treadmill and force them to run, they can then run,
Yeah, yeah. That's the next. I'm trying to hire a summer student to do those studies.
but if you just leave them like voluntary running, they seem to not want to run,
hmm
as much as their litter mates do.
Yeah,
So,
our hope was even if they ran a little bit, I didn't think they would run as much. The data is a little bit variable, but even if they run a little bit, maybe it would improve. That was kind of the hope. So
right.
we're not quite there yet. So
Well, thank you. It's interesting,
Mhm.
and multiple and multiple models may behave differently. So
Yeah.
there's inherent biological signal that comes
hmm
from the molecular defect as well.
Yeah. Awesome. Thank you.
Andrew, my name is Tae-hwa Chun. I'm an endocrinologist, and I'm colleague of Elif Oral, and I'm a basic scientist as well. So my question is, how many lipin or lipin family members you have in fat cells? And are there any report of those lipin-1 variants found in
hmm
human diseases or lipodystrophy?
Yes. So, Karen Rui's group at UCLA did a lot of this work. There are three lipin genes. And so the thinking is in humans with the lipin-1 mutation, 2 and 3 compensate. They're really not expressed until you have a mutant. And I think it's because this process, converting this into that, different fats is so important that there's redundancy involved. My advisor was studying this in everything from muscle to heart to liver. And it was kind of a side project to look at it in adipose, and that's how I got involved. But it's led me, to get a job in a K award, so I'm very excited about that. And it is a model. It's not the best, but we're learning and we're trying to adapt new ones. But yes, that is the thinking that humans compensate. And even in my mouse models, the other two, their expression increases. And this is in just went out in JCI, I guess a little bit a while ago. So you can read that if you want more.
Andrew, one other thing that I will mention, sometimes some things happen by faith, right? Like I'm
hmm
a firm believer. Just last Thursday, when we were reviewing one of our cases in our Radiant grant, there was a Lipin-1 variant
Oh, okay.
in a family, actually.
hmm
So we don't know exactly what that means. Obviously, they're at this point VUSs, but um, so that I mean, it just is something that we encountered. So
Very interesting. Yeah.
Andrew,
And for
in
the
one or two,
yeah, go ahead.
go ahead, Dr. Oral, go
No,
ahead.
I was just saying for those who don't know what RADIANT is, it's a network study funded by the NIH to recruit patients with rare and atypical forms of
Mm,
diabetes and patients with lipodystrophy who do not have a known molecular etiology are recruited into the study. So just a reminder, and I may have approached patients who come to U of M, talking to them about the RADIANT study. So just a reminder to our community that, we may approach you, or you can also approach us to participate in RADIANT. The application is actually online, so you don't have to come here to initiate participation.
Yeah, if you need help to get into the Radiant study, you can always email us, and we will definitely help. Andrew, just to finish up, can you tell us in like 2 sentences, how does your work will affect lipodystrophy patients in the future?
oh,
Just to summarize everything.
yeah. So I think, you know, I think the current therapies do the best they can, right? And so I think, we have, and I didn't talk about it today, the mice that we sort of I guess, make their adipose tissue dysfunctional, they will actually grow it back if we stop giving them the drug, right? And so, can we learn something about regenerating adipose tissue, and how does that, recover their metabolism and things like that? So I know there was some questions about that earlier. But there's been a lot of work on that, and so I definitely think there's room to grow, and I think that like I said, what is there? I mean, you guys would know more. There's what, one therapy? It's a very famous one, but maybe there's others, and maybe just learning how adipose affects the rest of the body, we can develop new therapies that are kind of not necessarily focused in the obesity realm, right? And maybe they're more focused on the lipodystrophy side as well. So I guess that hopefully that answers. Yeah.
Yeah. Thank you so much. And any other
I
questions,
guess if I
final
could
questions?
say, I know that the story earlier was very inspiring, but I think I just want people to know that there are a lot of scientists out there trying, and trying to understand these diseases, and really the exact reason why it happens, and what we can do about it. So um, keep that in mind.
Thank you for sharing
Mhm.
that. There's a lot of great people working on great things,
Mhm.
everyone. And some of them are on the call and some of them are all over the place.
Mhm.
So there's a lot of hope for our community. There's a question in the chat. Maybe anyone can answer this, any of the experts on the call. So those without adipose tissue likely have low leptin. Can you all explain exactly why?
I can start if you guys want. So
Yeah.
the organ that makes leptin in the body is the fat tissue. So if the fat tissue is not there, then leptin is not made. So it's similar to having type 1 diabetes uh, with insulin deficiency, right? In type 1 diabetes, the cells that produce insulin in the pancreas are destroyed or gone, and then you have low insulin. In some genetic forms of diabetes, the cells making insulin do not form, due to genetic programming. And then you have insulin deficiency and it leads to diabetes. So it's like that. When you have lipodystrophy, you in a very simplified model, especially generalized lipodystrophy, you have the fat cells either destroyed in acquired lipodystrophy, or they're they genetically do not form, and thus, uh, the result is having no leptin. In the different forms of partial lipodystrophy, depending on how much fat you have remaining in the body, your leptin level may be normal, it may be low, or it may even be on the higher side. It all depends on how much fat tissue is left or is there in the partial forms of disease.
Can I ask a follow-up question?
Sure.
So just, you used insulin as an example. In type 2, you can become insulin resistant. And I know in some research has been done on leptin resistance. Do you know if that occurs in lipodystrophy? Has that been sort of analyzed? So even though you may have low, maybe you also have resistance to actually signaling.
I mean, I think it's always a possibility. In when we treat patients with um, pure generalized lipodystrophy, single gene
Mhm.
and genetic defects that lead to lipodystrophy. Um, the clinical efficacy makes us think that we don't see much in the way of resistance. In acquired generalized lipodystrophy, it's not a pure metabolic disease. There is immune dysregulation and or autoinflammation, which may change leptin resistance too because of that inflammatory milieu, which can happen in the midbrain as
Yeah.
well, right?
Yeah.
So there may be some acquired leptin resistance in acquired generalized lipodystrophy. And then depending on the extent of body fat that we see in patients with partial lipodystrophy, the same principles apply. The things that we see in general population, the residual tissue stress and
Mhm.
adipose overengorgement, inflammation can lead to leptin resistance just as much as you see in obesity.
Okay. Interesting. Thank you.
Any other final questions for Andrew? Okay. Kate, do you want to introduce our next session?
Yes. So this is a really exciting session because Dr. Chun and Dr. Oral work with a member of our community in a more personal way through a case study. There was just a great article published about it. Mallory, if you're ready to put your camera on. And so we're going to do a little panel to have Mallory, Dr. Oral, and Dr. Chun speak about that experience and speak about their work to really show how clinicians and patients can work together. So we're really excited for that.
Mallory, are you coming?
Hello.
Yeah. Hi.
So to start, Mallory and Dr. Oral, do you want to give us a little overview of how you work together?
Sure.
Mallory, take it away.
Dr. Rel and I have worked together since I was 12 years old, so for about 12 years. And we have always been in close communication with each other. She's been talking to my mom too. My mom is also in the chat here. Pam, hi, my mom.
Yeah.
And we recently started a new, well, not recent, it's been almost about 7 years on Regeneron, which there's an actual name for it. I don't know how to pronounce it.
Nipocalimab.
Yep, that one. And first we started, my mom and I went and saw Dr. Rel every Tuesday, and then we go every 3 months now. And we've just had such a great relationship with her. And yeah, she just feels like another mother to me.
So, I mean, I recall meeting Mallory when she was 11 to 12, right? And Mallory happens to be very close in age to my kids. So I think meeting her with her mom was, like, uh, made me feel all those maternal feelings as soon as I met them. They also have a very touching story. Mallory, do you want to sort of describe that part of you,
Like how I was diagnosed?
or how you were diagnosed, and um,
Yeah, I started showing symptoms when I was 11. My mom noticed my legs were super thin and veiny, and she thought I was just deficient in protein or I was super dehydrated at first. And so we went to my primary care doctor, and they sent us to a genetics doctor at U of M, and he sent us to Dr. Oral, and we met her, and she knew exactly what was most likely going on. And so we were very fortunate to be diagnosed.
well, I mean, we knew that. I mean, she had a form of partial lipodystrophy, but it did have atypical features, right? There were some features that were different than all the other patients we had followed, or like there were some unique aspects in her presentation. So we thought that it might be worthwhile to bring in the newest genetic technology, especially after we've tested for all the known things, right? And Mallory and her mom are really close, and they work really well together, and their love just comes to you, when you meet them. So I think part of my my luck has always been meeting great patients and great stories. And partly owing to the experience of having given leptin for the first time and seeing the remarkable improvement that had happened in our very first patient and in the first 7 or 8 patients that followed, sort of had changed me forever. So that's one of the reasons why, when we have a patient who has something different, or known therapies don't work, that somehow like starts a button in my brain, that we have to do something, and but we have to keep pushing the boundaries. And sometimes we're lucky, we can find the answer. Sometimes we're not. There have been patients who I followed for 18 years, and then we had our aha moment. So this, is a long journey, right? And we've been through a journey like that with Mallory, right? We met her when you were 12, and here we are when you're 24, when we think we have a glimpse of what's going on in you biologically, right? So it was a long road. You stuck with it. You stuck with me. You entrusted in me. And you did have a big unmet medical need. There were times in your life when you were in the hospital the entire school year, right? Almost.
Yeah.
So we felt like we had to do something different for your therapy, and that's why like the investigational drug from Regeneron had come to the table. Um, we uh, actually had treated Mallory with Myolept first, and then she developed a neutralizing antibody to Myolept, and that's when her metabolic situation really worsened, and we felt desperate, and that's when we reached out to Regeneron for their drug, and they were excited to share it with us. And Mallory was the first patient that with lipodystrophy that received it, and she's to date the patient who's taken it the longest in the world. And it's a big leap of faith, right? Like to take a medication that hasn't been given in your disease state, but she's responded really well to it. And that allowed Regeneron to sort of develop its program of mebevidimab. So it was kind of a win-win for both Regeneron and for the lipodystrophy community. And it's all owing to Mallory's acceptance and sort of trust to receive that drug. But we know that that's not the cure. We know that wasn't going to make the disease go away, or whatever process that had started the disease. It's like, again, treating type 1 diabetes with insulin, right? You know, we don't make diabetes go away, but leptin is a drug like that. And the newer leptin agonist, mebevidimab, is a drug like that. Or the tirzepatides is a drug like that. You know, it doesn't fix what's going on underneath. So that's when I think we're now lucky to have more detailed genetic testing at our disposal. And we've done whole genome sequencing on Mallory that gave us a link, right, Mallory?
Yep.
And
My children.
yeah. And then from there, then Dr. Chun came into the picture, right? So Dr. Chun is my collaborator to pursue some of these new molecular leads in the lab. We have a committee that looks over these findings with us. And then the committee gives us a blessing that, yeah, this is worth pursuing. And maybe Dr. Chun can tell a few aspects that the committee considers, when they make that decision, that the story is compelling enough to pursue. And then the stepwise incremental work, right?
Yeah, so Dr. Oral was actively pursuing that, of course, the reason or a genetic variant that might be causing that lipodystrophy or atypical diabetes. And she already had multiple gene candidates, including Mallory's one. And then I was lucky enough to be invited on board, then partly kind of sponsored by Kahoot Diabetes Institute to help kind of develop a translational program from clinic to the bedside. I'm also an endocrinologist. I'm seeing patients with Dr. Oral together as well. So kind of it worked out well. And during that meeting, then there are several candidate genes. Of course, famous one is lamin AC. It's already well known. Of course, kind of gene variants to phenotype link, you still need to be studied more. And they already have the program going on with that. And there are several candidate genes. One of the genes was EBF2. It's an early B cell factor. It's transcription factor. And there is a stop gain mutation. That means protein will be truncated. Kind of shorter version of protein might exist in Mallory's case. And then we wonder whether that is the case. And this shorter version of EBF2 protein is causing the problem in her body. So that was the question we are asking. And the committee think that that's worthwhile pursuing, and because it's a kind of new gene. But the EBF2 itself has been known in mouse world, mouse adipose tissue, fat tissue biology. And especially there is a fat tissue called brown fat. It's a thermogenic fat. That means kind of increasing your body temperature. And people think that brown fat is good for your metabolism. Then there's a famous, several good researchers doing that work on EBF2, but nobody knows about EBF2 in human disease and human fat tissue biology. Then that was a starting point. And then at that time, Dr. Maria Freitas, she was working with Elif, and Then she volunteered to work with me as well, and then try to do using that gene variant, and then do the in vitro cell culture study. And we started from adipocyte differentiation model. Then we introduced this Mallory's variant versus wild-type BBF2, and we see a huge difference in terms of adipocyte differentiation, fat cell differentiation. So that is kind of first hint given to us. Then kind of we accumulated several data based on that. Then kind of we convinced committee, and so kind of maybe we should make it a mouse model so that we can understand better how this is causing lipodystrophy-like phenotype possibly, and also metabolic deterioration in Mallory's body. Actually, first, yes, then we created a mouse model, and putting in exactly same variant in the mouse using CRISPR-Cas and Queen model. Then they are born normal. We don't see any differences at the beginning. But later, if we put them on high-fat diet, it's kind of putting stress on your body, and they have to expand their fat tissues, right? Like we do in our body as well. Then in that case, they start to develop like scar-like tissues. It's called fibrosis, scar tissue in fat tissues. And similar, that is causing more problems to the body. Then before that, Maria already get some surgical sample from Mallory's surgery. She had like a lower back surgery also. Then we knew that she has scar-like tissues in fat, and also kind of strange appearance of that scar tissues. And we are always wondering what these are. Then we created mouse model. In that mouse model, especially homozygous. Homozygous means having variant in both allele, double the allele. Then Mallory's case is only one allele variant. But both allele variant is really sick, and their fat tissue looks really filled up with the scar tissues. And exactly the same scar tissue appearance we saw as we could see in the Mallory's surgical samples. That was as Elif mentioned, aha moment. It's maybe something is really happening. This variant is really causing the problem. Then maybe we decided to study more of this mouse model and using dietary modification or tissue histology or so, and then did the study.
Mallory, you disappeared on us. I hope we're not boring you.
Yeah, I was going to ask you, Mallory, what was it like to work with
I had to get water.
Okay.
What was it like to work with your doctors in this way?
Well, to be honest, I'm not really sure like what is the other type of way to work with your doctor, since I met them when I was young, and so this is always what I've known. But talking to other people, I know I have a really special privilege of having these close relationships with my doctors, and I feel very supportive. I never feel like I'm not being heard, so I'm really grateful.
We are also very thankful to Mallory. Mallory volunteered to provide additional fat tissue for biopsy as well. Actually, we are behind the door waiting to get the samples. When plastic surgeon working with you, and Dr. Maria Freitas was working with you. And another first author is called Donatella Giglio. She's from Italy, and she gets PhD based on this study. But I and Donatella was waiting in the behind the door to get tissues from you. And then actually that additional study, getting histology of Mallory's tissue, helped us to really matching that phenotype we saw in the mouse with Mallory's fat tissue appearance. Yeah.
I should also add that Pam, Mal's mom, right, volunteered to give her tissue.
Yeah, that's right.
Control.
That's yeah,
yeah, so we're always thankful to anybody, for undergoing a biopsy study just in the name of science. We're so grateful that our patients are willing to do this. There are others in this chat who've who've done it, and both normals and patients. I mean, you're basically accepting to be cut for in the name of science. That's so valuable. And, it's really hard to express the appreciation we feel. And the scientists and the students and the postdoc trainees, who handle your tissue, they're all at awe of that dedication. You inspire them every day because you are able to give your tissues like that. They get even more desire to do their work and to find answers for you. I mean, this is really incredible energy, in my opinion, right, Dr. Chun? You agree with me?
Yeah, I agree. We always think that science is usually driven by curiosity, and many people do the science for that reason. But of course, basic science is driving that revolution, transformation in medicine as well. But if we have the case like Mallory's, we are more connected and we can develop like a sense of purpose, or also your case, sense of urgency as well. And also easier for us to collaborate. But let's say totally different field of the research we are doing. Maybe we don't talk so much if we didn't have you. Then Mallory is there, then Elif Oral has lots of passion to pursue, and she is willing to reach out to many people. And then because of her, we come to the same table, maybe discuss about your case, and we come up with a plan. And then we start trying this and that to begin with. And if it's positive data we get, maybe let's try this or that. It's a really driving force, your relationship between Dr. Oral and Mallory's. Yeah.
No, I mean, and again, it's Mallory's curiosity and attachment to the science, has been inspiring everyone too. I think Maria had, brought up the point at one point. Mallory, I mean, you thought like the mice. I mean, she was asking us about the mice, like how many, after we had the founder line, right? Like how many pups did they have, and like how are they doing? And like sometimes looking at pictures of the mice, whether we were going to get a sizable colony, right? Mallory, I think you were asking us and feeling some ownership about it, right? About them. And I've seen this in other patients. Again, we've been lucky enough to create a number of human variants into mice. Or, we've also started to do Drosophila model, fly models. I think patients really get excited to know about their own conditions models, and they want to hear what's going on in the lab. And Mallory also did like a GoFundMe campaign for us,
Wow.
right? At one point, this project happened without real NIH grant behind it, right? That whole publication.
Still not.
It just survived on, scrapings of research dollars, from some savings account, or Caswell Diabetes Institute gave us pilot and feasibility money. And then our patients gave us money. And, I cannot thank Pam and Mal enough, to support our research this way.
Mallory, what would you say to people in our community or people on this call who want to get involved in science, but maybe have some reservations or fears?
I would say just ask questions at doctor's visits, even if it doesn't seem like personal to like what you would know. I don't know, just ask them anything. Any doctor would really like to go into what they're doing and the science behind it. And yeah, that's pretty much what my mom and I were just asked probably too many questions about what's going on. Dr. Rel's probably annoyed with all of our questions.
No, never. Never.
What role did trust play, Mal, in your work? Did you develop trust for your doctors over time? Do you feel like it was developed right off the bat?
Yeah, I definitely trusted all my doctors right off the bat. I have never heard of lipodystrophy when I was diagnosed, so I had no idea what to expect or what to even look out for. So, yeah, and I think it works both ways. Like she trusts me and I trust her, and that's how we have created such a great system going on.
Yeah, Dr. Oral creates a beautiful trust right from the bat. As you all know, she's kind of, and I haven't talked to Dr. Chun. I'm sure he's the same. It's kind of like
I'm not sure. She's better doctor, but
this beautiful friendship, I think, builds between the doctor and patient. And I think that's so special because
That's right.
it's not just about caring for you, Mallory. I know that Dr. Oral cares for you as a person, very truly. Pam, I see that you unmuted. Did you want to say something?
I did. From a mom's point of view, I lost my husband when I was pregnant with Mallory and her twin brother. And when we found out that Mallory had this lipodystrophy and we met Elif, I almost felt it as almost a co-parenting with Elif because I was uncharted waters. I was very afraid. I didn't know what her future was going to be, and Elif was very comforting to me, and we're still to this day very good friends. But I feel like I mean, how many doctors can you call at 11 o'clock at night because your child is screaming in pain and you need help, right? But I'm just very, very grateful that she has walked alongside us, or we've walked alongside her, and brought Mallory to this point. And I'm so excited for this research, and so grateful to everybody that has done so much and has worked above and beyond for lipodystrophy and all of these people here. And I have a lot of hope, and I'm just very grateful. So thank you.
Well, I am very grateful to have had the opportunity to get to know you and Mal and to work on this question. And truly, every patient who has come through our doors, I always say it's for a reason. So whatever it is, whatever that reason is, and whatever it is that we can do together, we will do. And if there is a stone that we can turn, we will turn it, and we'll find a way to turn it together. We never take no for an answer, right? I mean, I think that's what Tae-hwa knows, right? Like science is a lot about knows, and Andrew will probably know that too, right? Like we keep writing grants, we keep asking for money.
Yeah, it's expensive.
Yes,
It's really
but we will
expensive.
never take no for an answer. If we have to go somewhere, we'll go. If we have to get a little bit more money to keep alive, that project, we will. But it's because, you give us the reason for it. Knowing you all, knowing your struggles gives us the reason. So you inspire us every day, all of you.
And also, I think that thanking to that advancement in the technology, now we can do the next generation sequencing and the exome sequencing, and it also became more easier to collaborate with others. And if collaborated with the Harvard group and to do the gene analysis for Mallory, and that was a starting point. And then Casual Diabetes Institute enhancing the collaboration among researchers, then that is, and providing some startup money that really helped us as well to get started for Mallory. Yep.
So I want to bring you a little bit back when you decided to do the whole genome sequence on Mallory. I know several people on this call that have some kind of a genetic lipodystrophy, but they don't know the mutation. So my question to you is how those patients can be involved in such research, such as Mallory. Should they come to you and ask for this extended genetic testing? Should they go into a clinical study to do it? What's the way to get this kind of information for them?
So, if I could start, whole genome sequencing has become part of clinical care for potentially genetic etiologies now. And the access to it, at least in the United States, has widened a lot. So, if the diagnosis of lipodystrophy can be made and substantiated, there are ways your doctors can request clinical genetic testing and can request even clinical whole genome testing. These are done more readily, like in specialty centers, what we call like third tier referral centers. It's a little easier, but clinically available and accessible far more than before. If the clinical tests will report only known things, right? For unknown things, one has to be at that border of care and research. And then, like I think seeing a specialized doctor with a research group, that's where the difference might be. And we're lucky to have built that research infrastructure now. So where normal clinical testing it doesn't give us results, we could sort of push the frontier far better because of our infrastructure. And there are number of centers that have it. I think Kansas Children's has like a special program for children with genetic disease, and I've actually had referrals come to us from there after they've done the molecular diagnosis. So many medical centers are building this. In terms of research, in the United States, Radiant is one big resource where we could not only get whole genome testing, but we can also do additional gene expression tests from your circulating blood cells, PBMC transcriptomics, and bring those two together to find novel things. It does require, a lot of multidisciplinary discussion with the geneticists and the clinicians to sort of suspect a finding. All of us have, anywhere from 50 to 100 or even more unique variants. So how would you suspect a unique variant to be disease causing or disease contributing is sort of an interesting process. And I think to the extent that we can bring multidisciplinary interactions with geneticists and clinicians together, and even researchers, who have the expertise in labs together, that's how we push the boundaries. Do you agree, Andrew? And
Yeah, yeah. I don't have too much experience with, I'm not a geneticist by training, but I did want to say that, having human samples in the lab just kind of brings everything to a whole nother level. And so we really do appreciate that because we understand that it is kind of a sacrifice that you make, but it really, because I have a collaboration going with OR surgeons who give me adipose tissue. Granted, all of those patients say take as much as you want. But it definitely adds to like it just puts your research on a whole nother level and it just puts your purpose at another level. So I just wanted to reiterate that. Um, so
and Dr. Chun, do you have anything to add to availability of genetic testing and how we go about the discussions? I think
I think Elif covered well. So kind of if you were referred to Elif's clinic or so, then usually they run that clinically already certified lipodystrophy panel. Those candidate genes already known to cause lipodystrophy. And then you will work with the human geneticist in their clinic as well, and they can review all those. And if those are negative, then there might be something going on beyond that, those known genes. Then further, we will send a sample to that gene sequencing, either exome sequencing or whole genome sequencing. I think will be the discussion, as Elif mentioned, between clinicians and the geneticists. And then if there is resource available, maybe, and the RADIAN study is a big one, kind of they might offer that genetic testing as well. So I think there is always a way to get the gene testing done. And even if you pay out of pocket, it might be less than $1,000, I think, nowadays. And so I think there should be always a way. I think you should continue to talk with the clinicians and then kind of reach out to the regional center or sign up for those radiant study or so. I think there is always a way to get things done. Yeah.
I think there's a question in the chat from Omar, right? From Cambridge. He's saying, what's the turnaround time for patients to get their genetic results? In the UK, it's usually a few months. Wondering if faster for you guys. So if we are doing for just the known lipodystrophy panel, that comes back fairly quickly, 3 to 6 weeks. Whole genome may take longer. And if we're doing sort of like a whole pipeline review, that in the research world, to line up all the data and to get everything the way we want it, and then to have a meeting about it, it takes about anywhere from like 12 to 24 weeks. That might be long. And also, sometimes because that's in the research world, if there's a holiday or something, there might be a break. And in terms of giving the research results back to patients, there's a whole process for that. So it may not happen as quickly as we want in terms of coming back and telling us, our patients, what we found in the research world. But all of this is very rapidly evolving. The technology is really pushing forward fast right now. So, I mean, I won't be surprised if, like, within the next 5 or 10 years, probably more on the order of like 5 to 7 years, we come to a point where getting your whole genome sequence, just sort of like getting your whole body MRI done, like if you wanted a real thorough checkup, that might be something that you can have access to. I won't be surprised if we get there far faster than it seemed like it was going to be.
On the quick search, the lowest cost is $3.99 now for whole genome sequencing.
Yeah. The costs have come down enormously, again, in my lifetime in research, so, and how we could do this is pretty amazing now.
Yeah. Dr. Chan, finding Mallory's mutation and taking the approach from bed to bench, can we say that this finding will help Mallory someday? Or can you maybe tell us a little bit about finding the mutation? How can it help the community?
Yes, that's a goal. So kind of now knowing that this gene variant is causing the problem, then we will understand how this variant is changing that biology of fat tissues. And of course, if we can correct that first starting point of the gene variant, that is the best. But sometimes it might be challenging. But by knowing that downstream pathway, maybe we can bypass that problem, try to help her better as well. And in the mouse model, kind of we did the diet modification. Then high-fat diet is really kind of aggravated that histology, kind of more like a scar tissue, like a formation, and also metabolic deterioration. So But even though these mice having those gene variant, if they stay on the low fat, low calorie diet, their phenotype, their outcomes are not so bad. So they don't develop too much of diabetes, like on high fat diet. So that is still kind of one thing we can learn. Even you are at the genetic risk, by changing diet, maybe we can prevent or that worse outcome of that. And also, there's now we are starting some collaboration RNA therapeutics here at the University of Michigan, kind of using antisense oligo, whether we can diminish that bad toxic protein production from that gene variant. So if we can target that allele only, or another way is also there is other EBF family members. There is EBF1 and EBF3 in fat tissues. Maybe even if we knock down totally EBF2 in adult body, maybe those EBF1 and EBF3 can compensate for that. Then it might be better way than having toxic protein. So then knocking down both, then zero protein should be better than having toxic protein. That is another approach we are thinking as well. And there's some innovators in the field. There's some technology called PROTAC, or that is kind of trying to target that mutated protein only or a truncated protein only, and then to degrade them intentionally. So there is several researches going on. So we have to keep eyes open and maybe reaching out to those people who are in the different field and to see whether we can come up with a new plans for Mallory.
You're talking a lot about knocking in, knocking out. Can you talk a little bit about genetic therapies and how knowing the mutation might help us in the future with genetic therapies?
Yeah, so one of the famous ones is sickle cell anemia patient who received the stem cell therapy after the CRISPR-Cas gene correction, right? So kind of Mallory's case, or in general lipodystrophy, I think that the cell population in fat tissue is called kind of fat tissue stem cells. Then kind of when we are eating or so, kind of we are always renewing our tissue. We are making new fat tissues. But in that case, those stem cell-like populations are going into the fat cells. And we should be replacing old fat cells with the new healthy fat cells. But those people having some gene variant, usually those stem cells cannot differentiate well into the new fat cells, and rather staying like fibroblast-like cells, kind of producing more extracellular matrix is called scar-forming proteins. So whether we can change that with using those gene correction in the tissue level, or transplanting some newly healthy adipose stem cell, fat tissue stem cell, can be the possible things, maybe kind of possibility that we should explore as well. And how much healthy tissue, fat tissue you need for our body, nobody knows, right? So that's another thing. Maybe even small amount of healthy fat tissue or fat cell might be good enough to produce enough leptin, or another good hormone called adiponectin, or and also kind of interaction between healthy fat tissue and Andrew has been working on like a liver or skeletal muscle or heart. Those are important research topics as well. Many people are working on. Yeah.
Thanks. Any questions from you guys?
One thing I want to just add here, just again to bring the hope back, right? The doubling time of new information has really shortened, and it keeps getting shorter and shorter. So I think, we're in this era of really rapid transition and accumulation of new knowledge. So I'm really hopeful that we're going to get to precision therapy of genetic diseases fairly soon, right? Tae-hwa, I think 5 years is what I keep saying. I told my team we had to do this before 2030. So,
Yeah.
so we will try to stick to that time limit.
And AI-driven science is
Yeah.
expanding, so kind of they can also provide more opportunities and then even different approaches or kind of designing of those gene oligos. Also, kind of I think there's acceleration of that knowledge development and the treatment development is really amazing, I think. I agree with that.
Yeah, I just I want to add, when I first started my lab about 3 years ago, I spent $30,000 on this machine to identify gene expression, but it could only do one at a time. And now it's almost obsolete because we can do sequencing and get all of the genes at a much cheaper price. So it's definitely evolving very quickly, and I agree. I think just in a few years, these kind of things will be more routine, and the cost is going to go way down. So on the flip side, though, it is a lot more information. And so, like you said, my lab is actually using AI to help us manage all that information, but in a good way, right? So sometimes I think AI is good for that, but you have to be careful. So anyway.
I think it's exciting times, all in all. Despite all the negativity and conflict that exists in the world, I think thinking about, all of you and how we could change lives, and imagining that future, I think, like we have to keep the hope alive and really, I feel very excited about what can happen in the next 4 or 5 years.
And I really praise Elif as well because she is catalyst in a way. So because without her, those people in the different field of research kind of staying inside of, and she is willing to reach out to anybody to find a solution for patients. So that is, I think, the driving force for the collaboration for patients with lipodystrophy.
It's only because everyone's like generously giving their time, their energy, and then patients are giving their tissues, so like and their life stories to us. So we have to take that with the value that it deserves, everyone's contribution.
Thank you so much to Mallory, Dr. Chun, and Dr. Oral. That was an amazing panel. We really appreciate you all being here.
Thank you, Mallory and Pam,
Thank you.
for being here. And again, one more time for giving us a story to pursue.
Thank you.
So we have one last part of the day. A couple people just filtered in to do this final part. To wish everyone a happy almost World Lipodystrophy Day. We are going to do a little game, a nutrition game. Annabella on Dr. Oral's team helped me create these questions. We're going to do a little Kahoot, if you've all done a Kahoot. So if you have a second device near you, like a phone, I'm going to have you all scan a QR code to get into this game.
Oh,
so I'm going to go
I'm so bad at Kahoots. I can't.
Does everyone see this? Oh, I have to start. This is a game on nutrition in lipodystrophy.
Where's this QR code though?
Is this music loud? So what you're going to do is you're going to scan this QR code, or you could go to www.kahoot.it and enter the game pin. So you can do either one. Okay, yay. So it seems to be working. I see people filtering in. The little like characters are so cute. I'll give like 30 more seconds for people to get in. And the winner of this Kahoot gets a 1-year membership to Masterclass, which is a cool app that has different learning opportunities. How would you describe Masterclass, Sharon?
It's short lectures or talks about all kinds of subjects. You choose what you're interested in, and then you have just a list of like talks from experts about it.
Yep. Okay, is everyone in that wants to be in the game? Okay, cool. Should I go ahead and start?
Yeah.
Okay. Let's start this game of Kahoot. World Lipodystrophy Day, nutrition. In lipodystrophy, why can triglycerides become very high?
We can't see the
We can't
answers.
see the
Can't see
answer.
it.
You can't?
No, we couldn't.
You can't see them on your device?
No,
No, we
It's
just
see
just colors.
we see shapes and colors.
Oh no. Can you see on the screen the options?
No.
No.
I just hit one just to see if it would show me, and it says a bit too swift or something like that.
Okay. Now can you see?
No, I
No.
just see a black screen that says Kate Stratton has started screen sharing.
What? Oh no.
So see, like I got an incorrect answer.
It told me time's up.
Now we
Oh,
can
get
see.
out.
Oh, now we
Now we
can.
can
now
see.
we can.
Start over.
Should I just start the game over, or let's just go to the
Yeah.
next question?
Or let's just go to the next one. Yeah.
Body fat does not distribute or function properly. Everyone has 0. Okay. Which meal pattern best reflects the balanced plate method? Can you see the answers now?
Yeah.
No?
Yes.
Okay.
We see.
Half protein,
We can
half
see.
starch, 1/3 vegetables, 1/3 fat, and 1/3 starch. Half fruit, 1/4 protein and 1/4 vegetables, or half non-starchy vegetables, 1/4 protein and 1/4 starch or fruit.
This is difficult.
Nice. Most people got it right. Half non-starchy vegetables, 1/4 protein, and 1/4 starch or fruit. Jen's in the lead. Which type of carbohydrate is most encouraged because it is richer in fiber and or protein? White bread, sugary cereal, beans and lentils, or fruit juice?
The music just gotten super serious.
Nice. Beans and lentils. I would have been scared if people put sugary cereal. What eating order may help lower the rise in blood glucose after a meal? Carbohydrates first, vegetables last. Dessert first, then protein. Fruit first, then starch, then protein. Protein and vegetables first, carbohydrates last. Most people got it right. Protein and vegetables first, carbohydrates last. Does anyone know how to explain why this helps lower the rise in blood glucose?
The vegetables and the vegetables contain fiber, and the protein is also a little bit less likely to cause the insulin spike versus carbohydrates or starches or sugar or dessert.
Good answer, Gretchen. I like your answer.
Because Dr. Oral told me to.
That works too.
And I think some protein can help you produce insulin earlier, even before carbohydrates hit.
Yeah, I mean that there's definitely exceptions, right, to the rule. But I think if you, I think the best is to do the salads first, actually, right?
Yeah.
Salads first, then protein.
Then protein, and then the fat. You know, like if you're going to have the healthy fat, that's when you need to have it. Then do the carbs last.
Nice. And most people knew that. Why are refined carbs and simple sugars a concern in lipodystrophy? They can raise blood sugar quickly and increase triglycerides. They prevent digestion of fat completely. They reduce appetite too much. They always contain too much protein. Yep. Nice. Everyone knew that. Which of the following is the best example of a lean protein choice? Bacon, beef, skinless turkey breast, sausage. Nice. Skinless turkey breast. Which statement about personalized nutrition in lipodystrophy is most accurate? Everyone with lipodystrophy should follow the same diet. Nutrition needs vary, so meal planning should be individualized. Popular diets are always safe if they are plant-based. Only carbohydrate intake matters. Yeah, nutrition needs vary, so meal planning should be individualized. I think Dr. Oral would agree that it's important to talk to your clinician or nutritionist about what to eat and how, because people are very different. Anything to add, Dr. Oral?
No, I think you know, there are some apps that you can use to track a lot of your own data if you were so inclined. You know, they help you sort of really do individualization or more precision nutrition these days. So yeah, I think everyone does have a unique both genetic and epigenetic makeup. I think one size doesn't fit all. And what you are able to adhere to is also important, right? Like what you have access to and how you can access different things. So it has to fit your lifestyle.
Jared is still at the top. Which of the following may lower the impact of starch on blood glucose? Eating it with soda, removing the skin, refrigerating starches like rice or potato, and eating them the next day, deep frying it. Refrigerating starches like rice or potato.
Why is that?
Oh, wait.
Sorry, I'm embarrassed to ask the question. Why is that important? Oh, next question.
Oh, sorry. I don't know how to pause it. Let's talk about it after this question.
Yeah, just go on.
Why might plant proteins be especially helpful? They contain no carbs at all. They provide fiber and are gentler on the kidneys compared to meat. They always lower triglycerides immediately. They replace the need for medication. Nice. They provide fiber and are gentler on the kidneys compared to meat. Let's talk about why refrigerating like rice and potatoes and eating them the day after would help. Dr. Oral, do you know the answer?
I actually don't know at the top of my head. I was just going to look it up.
Yeah,
Really.
I can talk about it.
Yeah.
When you refrigerate starch, when you heat them and then cool them and heat them again and cool them again, you create resistant starch,
Yeah.
so it's harder to digest and it lowers the glycemic index. So the glucose would be like the high would be lower.
I mean, I
Wow,
think
good to
like
know. Thank you, Cheryl.
if you eat potato without cooking, right? You know, that's also resistant starch.
Yeah. Yeah, that's only Andra that does eat potatoes without cooking them. So
It actually is like a there are some animal model studies that show that cancer risk is reduced, you know, if you do that.
I would rather not eat a potato without cooking it, but
Okay. In fairness, it's when I had severe hyperphagia and I was starving. I couldn't wait for it to cook. Thank goodness there's treatment and I'm doing much better now. But yes, I have eaten a near raw potato. That is a true story.
okay, next question. What is one reason alcohol can worsen triglycerides? It blocks protein absorption, it removes fiber from meals, it lowers blood glucose too quickly, it prevents the liver from breaking down triglycerides. Nice. It prevents the liver from breaking down triglycerides. If someone uses insulin and is at risk for hypoglycemia, what is recommended? Include consistent amounts of carbs per meal and avoid skipping meals. Skip carbohydrates entirely. Only drink juice when hungry. Eat one large meal per day. Yeah. Include consistent amounts of carbs per meal and avoid skipping meals.
Oh, Elif is on top.
Which statement best reflects the role of nutrition in lipodystrophy? Diet alone can cure lipodystrophy. Nutrition is unimportant if medications are prescribed. Nutrition helps manage complications, but medications are still often required. Only triglycerides matter, not blood glucose or blood pressure. Yes. Medications are usually still required, but it's a factor and it can help. This is the final question. Which fruits are described as having a lower effect on blood glucose? Bananas and mangoes, berries, citrus, apples, pears, nectarines, peaches, and plums, dried fruit and grapes, pineapple, and watermelon. Yes, berries, citrus, apples, pears, nectarines, peaches, and plum. Okay, let's see who's the winner. Nice, Mal. Third place. Jared, second place.
You know what? You know what?
He
though?
doesn't
Like,
need one.
no. It is. I think the award should go to Gerard and Mal. So
Yes, we'll send. You know what? We'll just send all three of you a free masterclass subscription for a year. Since we have a bunch, we got a grant.
no, well, as I said, like the next one on the list should get it then. Is that okay?
Yeah, Dr. Ralph, can't you do it in all your free time?
Yeah.
We needed some Turkish coffee for this today.
Yeah. We can we should have some muddy Turkish coffee, right?
Gretchen can tell you about it. Me telling her about it.
It was so great to see all of you. Happy almost World Lipodystrophy Day, which is on Tuesday. And we're so glad you all came. And this recording will be available on YouTube, and we'll share it so you can share with your friends and family too.
Oh, before we go, I'd also like to say I'm starting a lipodystrophy podcast, and I would love to have some of you guys on it. So contact me if you would like to be on it, because I think that would be amazing.
That's great.
Great. Nice, Mallory. That's awesome. We'll support you in any ways we can.
Thank you.
Bye, everyone.
Well, thank you, everyone. It was great to see you all. Thank you.
Thank you.
Bye.